Okay, so my first task is biology B pre-test. So here we can see an image. So use this diagram to answer the following 6 questions. So the questions are: label 6 represent an organelle that produce sugar molecules. What is it called? So mitochondria, Golgi bodies, chloroplast, and vacuole. So my uh, so the image, this is 6. Okay, so it is not vacuole and it is not chloroplast. Yeah, then Golgi bodies, mitochondria. So I will going for chloroplast. Okay, and the next question is: label 4 represent dash, which is necessary for dash. So which is label 4 is this. So this 4 is vacuole. Yeah, it is vacuole, and which is necessary for dash. So central vacuole, maintaining turgidity or holding nutrients. So I think central vacuole maintaining turgidity. Now label 10 is the internal layer of a substance that surrounds the cell. This is called dash. So it is label 10. Okay, label 10 is this. So label 10 represent it is the internal layer of the substance that surrounds the cell. Okay, so it is, I think, endoplasmic reticulum. It is endoplasmic endoplasmic reticulum. Endoplasmic reticulum. Okay. Now the next question is: the main function of smooth endoplasmic reticulum represented by label dash is dash. So the main function of smooth endoplasmic reticulum is, it is 2, which is protein synthesis, 2 lipid synthesis, 9 protein synthesis, 9 lipid synthesis. So the main function of smooth endoplasmic reticulum, I think it is, okay, let's see what is 2 belongs to. Okay, so 2 is endoplasmic reticulum, and let's see what is 9. Okay. 9 is this, so which is also smooth. Okay, so it will go for 9 protein synthesis. Okay, now next is label 5 is present in animal cell only, plant cell only, some prokaryotic and some prokaryotics only. So label 5, so which is label 5? 5 is this. Okay, so this 5 is present in animal cell only, plant cell only, some prokaryotic and some prokaryotics only. So I will go for some prokaryotic and some eukaryotics. Okay, yeah. And next question is: the organelle labeled dash will be in higher number in muscle cells compared to fat cell. Higher in number in muscle cells compared to the fat cells. So let it be what is 1 represent. Okay, 1 is this. Next it is 5. Okay, next it is 5. Okay. Then it is 8. 8 is this. I think it is yeah, it is a nuclear envelope. And 10 is this. So we'll go with the number 5. Okay, let's check it. Okay, next question is, the backbone of a polymer of nucleotide contain ribose sugar. What is this polymer? It is of ribose sugar. So I think it is RNA. So DNA is deoxyribose sugar. Polypeptide, I don't think it is correct. Lipid also, I don't think it is correct. So RNA may be the, because it shows which contain ribose sugar. So it is RNA is the right answer. Okay. Next question is, a DNA monomer is called a nucleotide and is made up of 3 parts: a nitrogenous base, a ribose sugar, and a sulfate group. Which part is correct? So it is a DNA polymer is called a nucleotide and it is made up of 3 parts: a nitrogenous base, a ribose sugar, and a sulfate group. Which part is correct? So nitrogenous base, ribose sugar, sulfate group, none of the above. This is completely incorrect nucleotide. So I will go for none of this. Yeah, none of the above. Completely in nucleotide. Yeah, it is completely incorrect nucleotide. Now the next question is, why is RNA not the main carrier of genetic material? RNA is single stranded, so it carry half the information. RNA is less stable than DNA. Uracil is toxic and is in high amount it is lethal. RNA cannot form long chains. So RNA cannot form long chains. I think that's the correct answer. Okay, because yeah, this is also correct. RNA is single stranded, but it only, this is wrong, carry half the information. So RNA is less stable than DNA. That is also I think it is invalid. And uracil is toxic and it is high amount in lethal, and RNA cannot be a long chain. So I will go for this. RNA is less stable than DNA. So next is, justify your answer for the previous question. Okay. Yeah, since RNA have on a single stranded, it is more it is chemically unstable than DNA. Also, also uh, RNA is less reactive ribose sugar. Okay. Use this diagram to answer the following 7 questions. Okay, so it is, we can see an image here, diagram here, so we can answer the next 7 questions. Okay. According to the strandedness of this molecule, what type of nucleic acid is it? DNA, RNA, DNTP, and NTP. So, according to the strandedness of this molecule, what type of nucleic acid it is? We'll go for DNTP. According to the types of bases in the molecule, what type of nucleic acid is it? According to the type of bases in molecule. So it is RNA. DNA, RNA, DNTP, and NTP. So I will go for RNA. Okay. Next is, what is paradoxical about this image? Okay, so let's check the image once again. Yeah, it is A delta epsilon T. Okay, so both both shows a letter nation of alphabets and Unicodes. Okay. According to your answer to question 11, what must be the final part of the molecule? Phosphate, nitrogenous base, and dash. So amino acid, a basic group, ribose sugar, deoxyribose sugar. So it is deoxyribose sugar. Okay. Next question is, according to your answer to question 12, what must be the final part of the molecule? Phosphate, nitrogenous base, and dash. Amino acid, a basic group, ribose sugar, and deoxyribose sugar. Use the following information along with the previous to answer the following 2 questions. After testing, this nucleic acid is found to be an entirely of a thriving and successful alien genome. Chaff rules still apply. A deoxyribose sugar is found in its backbone. What is this delta and epsilon must look like? A and T respectively, both A, U, and C, both C. So U and C. Next question is, what conclusion can be drawn from your finding? A genome that is comprised of single stranded DNA is as sufficient as double stranded. An organism genome needs C and G to reproduce. The type of sugar does not matter. This must be an exception to Chargaff rules. So I think this is the correct answer. Okay. Next question is, reverse transcription is a inverse process to the ordinary central dogma of biology. It is represented as dash and dash. Separate your answer by a comma, then a space. Okay, so reverse transcription, it is a what synthesis of DNA from a RNA template, right? So this can be represented by, it can be represented by it is a synthesis of DNA to an RNA. Okay, it's the specification is separate your answer by comma, then a space. Okay. Now the next question is, the inverse of the process described in the previous question is, the inverse of the process described in the previous question is RNA synthesis, ribonucleogenesis, transcription, DNA cleavage. So this reverse transcription means it is the synthesis of DNA from an RNA template, right? So it is okay. So here the above question, here it is RNA to DNA. Okay. So here the inverse of the process described, inverse of this reverse transcription is I think it is the RNA synthesis. Yeah, RNA synthesis. Which statement about protein is correct? A straight chain of amino acid is indicative of primary structure. A beta spiral is indicative of secondary structure. A protein bonding with another is indicative of tertiary structure. Quaternary structure is completely impossible. So about the protein, right? About the proteins. So I think option D is the correct answer. Okay. So use this diagram to answer the following 3 questions. Okay. A DNA template strand contains this sequence: 3GTA, TTG, GCT, GTG, 5. What is the complementary strand of mRNA? Okay, so here we can see some options. So I will go for reverse of this process, right? So 5 dash. Okay, I'll go for this. What is the most likely reason that the sequence in the question has been written in reverse order from the answer? So here we can see the options. So it is customary to do so. It is not customary to do so, but it is easier to understand it this way. The template strand is read 3 dash to 5 dash and mRNA synthesis 5 dash to 3 dash. It should not have been written in this way. So the question is, what is the most likely reason that the sequence in this question have been written in the reverse order from this answer? So I will go for this. The template strand is read 3 dash to 5 dash and the mRNA synthesis 5 dash to 3 dash. Okay. What protein sequence does this mRNA code for? Separate each protein with a hyphen. So what is the protein sequence does this mRNA code for? Hmm. So um, code so from so I think it is GUG. Okay, UUG. Okay, I will write it in the capital letter. GUG UUG AAA. Okay, so here we can see the next question. A point mutation has occurred but the polypeptide is same. What is this mutation called? Silence, nonsense, missense, intel. So here a point mutation has occurred but the polypeptide is the same. So I think it is missense. A point mutation occur in a separate polypeptide. The second one interact with the first one in an epistatic manner and cause the original polypeptide to become truncated. What type of mutation is the second mutation? So it is intel. Silent, nonsense, missense, and intel. You can just submit it. And I'm just checking my score. Okay, it is 8 by 25. So let's check what I have done mistake. Okay, so first question, chloroplast, it is the correct answer. Second question is okay. Second question, it is central vacuole and maintaining turgidity, which is also correct. Okay, which is also correct. So next is so here I have gone wrong. Label 10 is the internal layer of a substance that surround the cell. This is called membrane is the correct answer, not endoplasmic reticulum. Okay, so okay, next question is the main function of smooth endoplasmic reticulum represented by it is okay, correct answer was lipid synthesis. Okay, so let's check about this. So function of endoplasmic, yeah, reticulum in plant cell. So what is the function of this endoplasmic reticulum? Let's check. The endoplasmic reticulum transfers in two forms: a type with a ribosome studded surface and around with a smooth surface. The latter is called the smooth endoplasmic reticulum and the former is called the rough endoplasmic reticulum. So what is the okay, so here we can see this endoplasmic reticulum figure: rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum have ribosomes embedded within its structure, giving a rough appearance. The smooth endoplasmic reticulum does not have these ribosomes, hence appearing okay, smooth. So the main difference is rough endoplasmic reticulum has ribosome embedded within its structure. Smooth smooth endoplasmic reticulum, it does not have this ribosome, okay, which make it appear smooth. So next is the structure of endoplasmic reticulum. The structure of endoplasmic reticulum is shaped like a sac. Okay. Since this endoplasmic reticulum is of two types, each have its own distinguished features. So we know rough endoplasmic reticulum structure and smooth endoplasmic reticulum structure is there. So the rough endoplasmic reticulum is named as because of its performance. It is a series of connected flattened sac having several ribosomes on its outer surface, hence the name. It synthesis and secrete protein in liver, hormone and other substance in the gland. Rough endoplasmic reticulum is prominent in cells where protein synthesis happened. Okay. So smooth, what is the speciality of smooth endoplasmic reticulum? Because it does not have the ribosome. Yes. The smooth endoplasmic reticulum has a tabular form. It participates in the production of phospholipid, the chief lipid in cell membrane, and are essential in the process of metabolism. Okay. Smooth endoplasmic reticulum transport the product of the rough endoplasmic reticulum to the other cellular organelles, especially the Golgi apparatus. Okay, the smooth um, endoplasmic reticulum transfer the products from rough to Golgi apparatus. Okay, then yeah, here we can see the function. So the function of endoplasmic reticulum in the question is the main function. Okay, the main function. Okay, so the endoplasmic reticulum is categorized into two, and both of these having a specific function. So smooth endoplasmic reticulum is responsible for synthesis of essential lipid. Okay, so our answer should be yes, it should be lipid synthesis, not protein synthesis. So here the correct answer is D. Okay. So smooth ER is also responsible for the production and secretion of steroid hormone. It is also responsible for metabolism of carbohydrate. The smooth endoplasmic reticulum store and releases calcium ion. These are quite important for nervous system and muscular system. Okay, so the main function of the smooth endoplasmic reticulum is to um, synthesis of lipid, that is lipid synthesis. And we can check the rough endoplasmic reticulum function also. The majority of the function of rough endoplasmic reticulum is associated with protein synthesis. So the main function, the question asks for the main function of smooth endoplasmic reticulum. So for smooth endoplasmic reticulum, the function is lipid synthesis, but for rough, it should be protein synthesis. Okay. The rough endoplasmic reticulum also play a vital role in protein folding. Okay. Also ensure the quality control. The second most important function after protein synthesis is protein folding in protein sorting. Okay, is for protein folding as protein sorting. Okay, so here our option is lipid synthesis. Okay, and label 5 is present in some prokaryotic and some eukaryotics, which is correct. The organelle label dash with a higher number in muscle cell compared to the fat cell. So it should be of 5. Okay, 5. 5 I have marked which is wrong. So 1 is the correct answer, which is 1. 1 is this. Okay. 1 is the correct answer. Okay. The backbone of the polymer of nucleotide contain ribose sugar. It is RNA. Okay, which is correct. And DNA monomer is called a nucleotide, and it is made up of 3 parts: a nitrogenous base, a ribose sugar, and a sulfate group. Which type is correct? Which part is correct? Nitrogenous base is the correct answer. This is wrong. So let's compare the difference between DNA and RNA. Okay. Difference between DNA. Okay. DNA and RNA. Okay, so here 5 key differences and comparisons. Okay, so comparison DNA and RNA. So full name is deoxyribonucleic acid. Here it is ribonucleic acid, and the function of DNA is DNA replicates and store genetic information. Okay, genetic information, it store genetic information, and it is a blueprint for all genetic information contained within an organism. This is DNA function, and RNA convert genetic information contained within DNA to a format used to build protein and then moves it to ribosomal protein factories. Okay. About the structure, DNA contains two strands, whereas RNA it have only a single strand. Arranged here in DNA arranged in a double helix. Okay. This strand are made up of subunit called nucleotide. Each nucleotide contain a phosphate, a 5 carbon sugar molecule, and a nitrogenous base. Okay, so it mainly contain of phosphate, nitrogenous particles, and carbon sugar molecule, 5 carbon sugar molecules. Okay, now RNA only has one strand, but like DNA, it is made up of nucleotide. RNA strand are shorter than DNA strand, and RNA sometimes form a secondary double helix structure, but only intermittently. Okay. And about the length, the DNA is much longer polymer than RNA. A chromosome, for example, is a single long DNA molecule which would be several centimeters in length when unraveled. And about the RNA molecule are variable in length but much shorter than long DNA polymer, and a large RNA molecule might only be a few thousand base pair long. So here, as per our question, which part is correct, right? So here we can say that this nitrogenous base is the correct one. Okay, and we can go for the about the other comparison also. About the sugar, The sugar in DNA is deoxyribose, which contain one less hydroxyl group than RNA ribose. RNA contain ribose sugar molecule without the hydroxyl modification of deoxyribose. Okay. Now about the bases. The base in DNA is adenine, thymine, guanine, and cytosine, whereas in RNA is adenine, guanine, and cytosine with the DNA but contain uracil rather than the thymine. Okay. Base pair is AT, adenine, thymine, and cytosine and guanine. Here it is AU, adenine and uracil. Uracil, cytosine, and guanine. Okay. And location, this DNA is found in nucleus, and RNA forms in nucleolus and then move to specialized region of cytoplasm depending on the type of RNA formed. Okay. And reactivity due to its deoxyribose sugar, which contain one less oxygen containing hydroxy group. DNA is more stable molecule than RNA. Okay, DNA is more stable than RNA. So here DNA contains a ribose sugar which is more reactive than DNA, and it is not stable in alkaline condition. Okay. Next is ultraviolet sensitivity. Okay, DNA is vulnerable to damage by ultraviolet light. RNA is more resistant, okay, to damage from UV light than DNA. Okay, so ultraviolet stability is more for RNA. Okay, now let's check the another questions. Why is RNA not the main carrier of genetic material? Because RNA is less stable than DNA. So this is correct. Justify your answer for the previous question. RNA has one more hydroxyl, okay, one more hydroxyl group than DNA, so it is less stable. Okay, so the reason is RNA have one more hydroxyl group than DNA, which is make it less stable. Okay. Use this diagram to answer the following 7 questions. According to the strandedness of this molecule, what type of nucleic acid it is? It is RNA, not DNA. Okay, so it is RNA. Okay. According to the type of bases in the molecule, what type of nucleic acid is it? So it is also RNA. RNA have marked, so correct answer is DNA. What is paradoxical about this image? The single stranded nature indicate DNA while the type of base indicate DNA. So single stranded nature indicate RNA. Okay, single stranded nature indicate RNA and the type of bases indicate DNA. Okay. Next question is according to your answer to question 11, what must be the final part of the molecule? Phosphate, nitrogenous base, and it should be of ribose sugar. Okay, ribose sugar is the correct answer, not deoxy sugar. According to the answer to question 12, what must be the final part of the molecule? Phosphate, nitrogenous base, and deoxy sugar, which is correct. And about this question, here what this, okay, delta and epsilon must look like. It is U and C I have marked, so answer is A and T, okay, respectively. So adenine and thymine is the correct answer. What conclusion can be drawn from your finding? It is a genome that is comprised of single-stranded DNA is as sufficient as double-stranded. Okay. Reverse transcription is the inverse process to the ordinary central dogma of biology, and it can be represented as RNA and DNA. Yeah, this is correct. RNA to DNA, it is correct. Okay. So, next question is the inverse of the process description in the previous question is dash. So, it should be transcription, not transcription. It is not RNA synthesis. So, let's check about this transcription. Transcription and translation. Okay. Transcription is the biological process in which a copy of gene DNA sequence is produced and written into RNA. Translation means the biological process in which the protein is synthesized using genetic information contained in messenger RNA template. Okay, so let's check here also. So these are the initial step in assessing genetic information. Transcription is the first step in the decoding a cell genetic information. During this transcription, enzyme called RNA polymers build RNA molecule that are complementary to a portion of one strand of DNA double helix. RNA molecule differ from DNA molecule in several important ways. Yes, we have already discussed about these things. Single stranded, and the sugar component is ribose rather than deoxyribose, and they include uracil nucleotide rather than thymine nucleotide. Okay, yes. Okay, let's check the next one. Which statement about protein is correct? A straight chain of amino acid is indicative of primary structure, so this is the correct answer. Option A is the correct answer. Yes. So here we can see the next question. The DNA template strand contain their sequence. What is the complementary strand of mRNA? So it is 5 dash CAUAAC UU CGA 3 bar. Okay, so option 3 is the correct answer. What is the most likely reason in the question has in the sequence that the sequence in the question has been written in reverse order from the answer? So this is correct. Yes. What protein sequence does this mRNA code for? Separate it with hyphen. Okay, so it is HISASN PHEARGS. Okay, HIS. So let's check for this okay, mRNA code. search for images first. Yes. So it is about the codons and anticodes. So yeah, here we can see it. There are 64 codons are involved in the termination of protein synthesis and release the polypeptide from the translation machinery. These triplites UAA, UAG, UGA are called nonsense codons or stop codons. AUG is special codon as along with adding methionine during transcription. It also acts as the initiation or start codons. The process of this translation start from AUG codons, okay? Anticodes are the corresponding triplicate nucleotide sequence found on the transfer of RNA molecule, okay? Yes. So here, okay, so here a point mutation has occurred but the polypeptide is same. What is this mutation is called? So let's check about the different types of mutations. Different types of mutations. Yes, so type of mutations. Okay, so these are the type of mutations. There are mainly different way that the DNA can change, resulting in different type of mutations. So first one is substitution, insertion, deletion and frame shift. So let's check for, okay, so this is substitution, which means the original code is this, some of the letters replaced by another letter. So here we can see A is replaced with G, so here it is substitution. Insertion is additional, some codes has been added or some base pairs has been added. Deletion means some of the base pairs have been deleted. Frame shift is if we have shift the first base pair, what happened, everything has been shifted, so that it is a frame shift. But we can check for these terms such as silent mutation, nonsense mutation, okay, silent mutation, okay, silent mutation, okay, same, it is also called as same sense mutation and are mutations in DNA that do not have an observable effect on the organism phenotype, okay. That is silent mutation. So here as per the point mutation has occurred but the polypeptide is the same, yes, so which is silent. So silent mutation is the correct answer. So let's check for what is this nonsense, missense and intel means. Nonsense, missense, intel mutation, okay, 4 categories. Okay, so here missense mutation means the mutation that changes an amino acid in a protein, okay. And nonsense mutation means a mutation that changes an amino acid in the protein to a stop codon which ends synthesis of protein at that location, okay. And what type of mutation is intel? Okay, indel means the mutation named with the blend of insertion and deletion. It refers to a length difference between two alleles where it is unknowable if the difference was originally caused by the sequence insertion or the sequence deletion. So it is actually a combination of this insertion and deletion. Okay, so here our answer is silent because a point mutation has occurred but the polypeptide is same, which means it is a silent mutation. A mutation occur in a separate polypeptide. The second one interact with the first one in an epistatic manner and cause the original polypeptide to become truncated. What type of mutation is the second mutation? So it is, yes, it is nonsense. Okay, so we have uh, corrected 8 tasks here. Okay, so 8 tasks out of 25. Okay, so now we can check for the functions of functions of cell organelles. Okay, so here we can see the organelle function types and importance. Okay, so these are the functions of this organelle. Nucleus is to store genetic information. Okay, nucleolus, it is make ribosomes. Okay, cytoplasm contain the contents of the cell. Cytosol, gel-like matrix that hold water and nutrients. Cytoskeleton, it is a cell type is eukaryotic, structure, support and transport. So we can learn with this an image, right? Okay, so okay, yes, so ribosome, it makes protein. The function of ribosome is to make protein. Okay, and next is rough endoplasmic reticulum, which make protein for the endomembrane system. Okay, smooth endoplasmic reticulum, yeah, here we can see smooth and rough endoplasmic reticulum. So smooth endoplasmic reticulum is to deoxify the cell and makes lipid. Okay, Golgi apparatus. Golgi apparatus means this one. Okay, so this Golgi apparatus will sort and ship proteins. Okay, it will sort and ship the proteins, this Golgi apparatus. Okay, next is mitochondria. Yes, so this is mitochondria. The function of mitochondria is to make energy. It is for make energy. Then peroxisomes, it regulate biochemical pathway like that involve oxidation. Vacuoles store water and nutrients. Store water and nutrients. Lysosome remove unwanted materials and waste. Peroxisomes regulate biochemical pathway that involve oxidation. Vacuoles store water and nutrients. Vesicles transport materials around the cell. Cell membrane, it is a thin flexible barrier that separate the cell from its environment. Yeah. Next is cell wall. It is a rigid barrier that protect the cell. Next it is large central vacuole. It store water and regulate turgose pressure. Chloroplast, it's make food using the process of photosynthesis. Okay. So this is about the eukaryotic cell organelles. Yeah. So let's check the difference between animal cell and plant cell. Plant cell diagram. Yes. Okay. So this is a okay, yes, this image well shows the difference between plant cell and animal cell. This is the plant cell. Plant cell have a cell wall which is rigid in shape, whereas animal cell no cell wall, so that it is irregular in shape. Here plant cell we can see a large central vacuole. So in this image also here we can see a central vacuole. Okay. Here it is numerous small vacuoles. So here we can see small vacuoles are there. Yeah, small vacuoles, pink dots. Okay. Then chloroplast is there for plant cell. Here it is no chloroplast. Here lack centrosomes. Here it is centroles or centrosomes are there. Lack lymphosomes. So here we can see two extra things in animal cells, which is centrosome and lysosome. Okay. So these are the main difference. Then one more thing we can check this. Central dogma of molecular biology. The central dogma of molecular biology deals with the flow of genetic information within the biological system. It is often stated as DNA makes RNA and RNA makes protein. Okay, so although this is not its original meaning, it is first stated by Francis Crick in 1957. Okay, so this central dogma, this state that once information has passed into protein, it cannot be get out again. In more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but the transfer from protein to protein or from protein to nucleic acid is impossible. So here the transfer of information from nucleic acid to nucleic acid or nucleic acid to protein is possible from but it does not it is impossible to transfer the information from protein to protein or from protein to nucleic acid. Okay, so this is the theory behind central dogma or yeah, central dogma of biology. Okay. Yes. So that's it. Now let us move on to our post test. Yes. Okay. Here label G, which is G. Okay, G represent several protein that assist with cell division, protection and maintenance of cell shape. Okay. What does this protein comprise? It is cell body, cytoskeleton, head, musculoskeleton system. So I think it is cytoskeleton. Okay. Label F represent dash which is necessary for dash. So what is label F is this. Okay. So it represent, I think it is centrioles, and which is necessary for cell division, I think. Yeah. The organelle represented by D is mainly found in dash and dash. Your answer should be in the name of the organelle. Separate your answer with comma and then space. So I think it is rough endoplasmic reticulum, and okay, reticulum, um, and cytosol. Okay, it is only as D. Okay, next is the main function of Golgi apparatus represented by label dash, and it is lad dash. Okay. Golgi apparatus. Yeah, this is Golgi apparatus. So I think option D is the correct answer. The organelle label C is represented in C is this, right? So it is present in both animal cell and plant cell. Okay. The organelle label dash will be in higher number in muscle cell compared to fat cell. I think the options are A, C, E, B. Okay. A, C, E, B. So it is A. Let's check A. No, A is not the right one. C. No, C is also not the right one. E. Okay, is also not the right one. H is what? H. H is this. Okay. Next. Just as an amino acid in a monomer of protein, a dash is the monomer of DNA. It is nucleotide, right? Nucleotide. Okay, it is nucleotide. Next is a DNA monomer is called a nucleotide, and it is made up of three pairs: a nitrogenous base, a deoxyribose sugar, and a sulfate group. Which part is incorrect? So the question asks for the incorrect one. So deoxy sugar, yes, it is a part of DNA. nitrogenous bases also it is a part. Sulfate group, I think sulfate group is the wrong one. Yes, the question asks for which part is incorrect, so it is the sulfate group. Okay. Next is DNA is dash stable than RNA and therefore dash reactive. So DNA is more stable than RNA and it is less reactive, right? So more and less. So option A is the correct answer. Okay. Justify your answer for the previous question. So DNA is more stable than RNA and less reactive because because of this deoxyribose sugar have one less hydroxyl group. So because deoxyribose sugar has one less hydroxyl group. Riboxy sugar have one less hydroxyl group than ribose sugar. It is than ribose sugar. Okay, so that is a main reason. Okay. Use this diagram to answer the following 7 questions. Okay. According to the strandedness of this molecule, what type of nucleic acid is this? This is yeah, it is DNA. According to the type of bases in the molecule, what type of nucleic acid is it? It is RNA. DNA, RNA, DNTP and NTP. So I think it is RNA. What is the paradoxical about this image? So it is about the strandedness, right? Strandedness of the molecule is indicated of DNA, while the molecule contain uracil, which is a base, that is RNA. But RNA molecules uracil as base. Okay. According to your answer to question 11, what bases do delta and epsilon represent? It is A and D. According to your answer to question 12, what does 2 delta and epsilon represent? A and T, A and U, T and A, U and A. So I think it is A and U. Okay. Use the following diagram along with the previous to answer the following two questions. Okay. This is a sugar from the previously discussed nucleic acid. There is a hydroxyl bonded to the 2 dash, 3 dash and 5 dash carbon. What type of nucleic acid it is? So I think the options are DNA, RNA, DNTP and NTP. So here there is a sugar from the previously discussed nucleic acid. So there is a hydroxyl bonded to 2 dash, 3 dash and 5 dash. So it is belongs to RNA. Okay. What conclusion can be drawn from your finding? DNA can sometimes include uracil. The backbone of DNA can sometimes include this type of sugar. The amount of hydroxyl group does not matter. RNA can sometimes be found as double-stranded. I think the RNA can sometimes be found as double-stranded. Okay. Now the ordinary flow of information in the cell is dash, dash, dash. Separate each answer with comma then space. So the ordinary flow of information in the cell. So as we discussed in the central dogma of biology, it is from DNA, RNA, then to protein. Protein. Okay. The process that take place at the first arrow is dash and the process at the second arrow is dash. It is the process that take place at the first row, that is from DNA to RNA is transcription, and from RNA to protein synthesis it is translation. So let's check the option. RNA synthesis, protein synthesis, this is not the right one. Ribonucleogenesis and proteogenesis, transcription and translation, yes. DNA cleavages, codon anticodon interaction. So transcription and translation. So this is the correct answer. Okay. Now, next question is, which statement about protein is correct? An alpha helix is indicative of primary structure. I think this is wrong. A beta sheet is indicative of secondary structure. This is, I think this is correct. Yeah. Multiple proteins interacting is indicative of tertiary structure. Quaternary structure is only theoretically possible. So I think these three are wrong. So beta sheet is indicative of secondary structure is the only right one. Okay. So here a DNA template strand contains this sequence 5-GAG AAC TAT AGT CTG 3-. What is the complementary strand of mRNA? Okay. So complementary strand, let's check the options. So here it is, everything is between 3- and 5-. So it is GAG AAC UAU AGU CUG 5. No, that is not be the right one. 3 CUC, UUG, AUA, UA, UCA, GAC. Yes, so this can be a right one. Let's check others also. 3 dash GUG, UUG, AUA, UCA, GAC, 5 dash GUG, AAC, UAU, AGU, CUG. So I think this is the correct one. Your mRNA strand of choice must be reversed before converting to amino acid. That is, it must be written dash as DNA again, and complementarily to answer to the previous question as tRNA from 5 dash to 3 dash. I think this is the right one. Option B is the right one. Or from 5 dash to 3 dash. What protein synthesis does this mRNA code for? Notate each protein as a 3-letter abbreviation and separate it with a hyphen. Okay, so it should be GLN. LE okay. A point mutation causes an amino acid to change to a stop codon. What is the effect on the resulting polypeptide? So no change, truncated and non-functional, doubly effective, goes to nucleus to function. I think it is no change, it is not the right answer. Double effective, it's also wrong. Goes to nucleus to the function. So I think truncated and non-functional. Second point mutation caused that stop codon to change into a codon that is different from the original. What is this mutation? So this is a second point mutation caused that stop codons to change into codon that is different from the original. So I think this is missense is the correct answer, because Sidon is not that one, nonsense, and Intel is also not that one. So I think missense is the right one. Then we can submit it. Then we can check view score. Okay, we have 19 by 25. Yes, great job. So what all things were wrong? So actually this is rough endoplasmic reticulum and cytosol. This is correct, right? Okay, so here these RNA have more hydroxyl group than DNA, which make it less sensitive, less stable and more reactive. Yeah, similar to this. Okay, so here also the double stranded is indicative of DNA. This, while the presence of uracil indicate RNA. Based on these two pieces of information, it is inconclusive. Okay, so that is the wrong, where we have wrong. The ordinary flow of information in the cell is DNA, RNA, protein. So this is also right one. Okay. Okay. Anyway, we have scored better compared to that of our pre-test.